Well, let's talk about each one of these organelles in a little bit more detail. Starting out with lysosomes. Why am I going to start out with lysosomes? Well, I just finished doing all of that membrane material and bulk transport, so we just got done talking about vesicles and vacuoles, and here you can see some endocytosis happening in the cell, right? We got a vesicle starting to form, pinching off from the cell membrane, so we got some food coming in, forming a vacuole or vesicle, depending on how big it is or whether it's for storage or short-term transport, and a lysosome is approaching it. Now, a lysosome is very much like a vesicle. It is a membrane-bound little bubble inside the cell, but it contains digestive enzymes. It has lipases in it. Those are enzymes that break down lipids. It has DNases in it, enzymes that break down DNAs. Whenever you hear A's, that's an enzyme. It has RNases, it has proteases, it has carboases, it has ases for every kind of molecule that you could imagine. So it's able to break down all kinds of things. Since it can break down just about any kind of biological molecule, and of course the cell is made up of structures which are themselves built from biological molecules, you can imagine why you want to keep those enzymes under lock and key. So we bind them up inside these lysosomes. And since the cell is bringing in nutrition in through these vesicles, well, we have all the food in their own vesicles, and we have all the digestive enzymes in these kind of vesicle-like structures, these lysosomes. So how do we get digestion happening? Easy. These are membranes, right? Liquid membranes. And the great thing about membranes, you can push two membranes together and they merge. So when we push them together, their contents, their interior contents spill into each other. So we get the enzymes, the digestive enzymes, spilling into that food vacuole and breaking up little bits of starch or whatever it is we just took in, or maybe it's a bacteria that we're breaking down, and cutting it into its component pieces. We're seeing some hydrolysis reactions taking place. Earlier in the chemistry section, I showed you dehydration reactions and hydrolysis reactions where we added water and took it away in order to build up a polymer or break it down. And that's still happening here, but now we're adding the extra little intrigue in biology, which is that pretty much every chemical reaction, everything that happens in metabolism, is catalyzed by one or more enzymes. You don't really get any kind of chemical work done inside a cell without an enzyme helping you along. So those hydrolysis reactions that are used for digestion, enzymes are going to be helping it along, and those are going to be largely contained inside your lysosomes. Oh, hydrolysis, lysosome, lysis. You remember that word, right? We had cells lysing when they brought too much water inside, when they were placed in a hypotonic environment. Lysis is to break. Hydrolysis is to break with water. Lysis is for a cell to burst open because it brought in too much water. Lysosomes are breaking bodies. Some means body. So a breaking body, it is a little stomach that floats around inside the cell looking for a vesicle to merge with so it can begin digestion. Very cool. In this micrograph over here on the right-hand side, you can see a lysosome and it's starting to digest a broken organelle. This is a mitochondria that it's going to take down into constituent parts. So lysosomes can break down bacteria, especially when we're talking about white blood cells after they engulf some bacteria. They can break down broken or damaged organelles and reuse the parts. They can break down food and get nutrients out of them. And then once we break them down into components small enough, those components, those monomers, can diffuse out of that cell membrane and return back into the cytosol, so we can use them to make new types of molecules. So this is how we get nutrients into the cell. This is how the cell essentially digests its food. In some cases, lysosomes can play a role in programmed cell death, where if you rupture a lysosome, all those digestive enzymes can eat the cell from the inside out, and that's kind of a self-destruct mechanism for a cell, and that can come in handy in certain cases as well, like if the cell happens to be infected by a nasty virus and it knows that it's not long for this world, well, rather than allow it to keep propagating viruses, sometimes a cell will undergo apoptosis and programmed cell death, and it will rather self-destruct than continue on. So lysosomes are a relatively small organelle, but a pretty big deal nonetheless. Next up in our tour of organelles, we have vacuoles. Vacuoles are large storage containers inside of the cell, so bigger than a vesicle. Vesicle is more for transporting material around the cell. Vacuoles more for long-term storage. And we've already seen food vacuoles and that sort of thing. And we've talked about the central vacuole as well. So you guys know that the central vacuole inside of a plant cell, which you won't find in an animal cell, is going to store water. Sometimes there's going to be some nutrients in there and may have some pigments or poisons mixed in, depending on what kind of plant we're talking about, what kind of leaf it is, but mostly it's there in order to maintain water pressure inside of the cell. But there are other types of vacuoles as well. So this is a paramecia, and paramecia sometimes have what are called contractile vacuoles in them. And this is what the contractile vacuole is. This paramecium lives in a freshwater environment, so its cells are hypertonic to the environment. It's got more solutes inside of its cells than the environment. So it's always taking on water, but it doesn't have a cell wall to prevent it from taking on too much. So you think, oh God, it's gonna lyse. It's gonna be like that red blood cell we talked about in the other video. It's gonna burst open. But here's what it does. It takes all of that water and it gets shunted into this contractile vacuole. And the vacuole just keeps expanding and expanding and expanding and expanding. And then a ring of proteins squeezes all of that water on out, and then more and more water keeps rushing in, and it keeps expanding and expanding and expanding and expanding, and then it squeezes that water on out, and it's able to maintain its internal osmolality, the amount of solutes that it keeps inside without lysing and without becoming diluted or anything like that, just by using this contractile vacuole system of continuously filling it up and squeezing it out, filling it up and squeezing it out. This is the microbiology version of being in a rowboat with a hole in it and using a bucket to constantly bail the water out, bucketful by bucketful. Water's coming in, but you're just continuously scooping the water out and tossing it over the side. And yeah, it seems like it would be a lot of work, but hey, if it keeps you afloat, it keeps you afloat, and the contractile vacuole seems to do its job. Next up on our tour of organelles in the cell, we have the nucleus. The nucleus is frequently referred to as the most important organelle inside of a eukaryotic cell. I mean, the word eukaryotic is referring to the nucleus. It kind of means true nucleus. It means true seed or true kernel, but it's referring to the nucleus, so it's true nucleus. It is the storage container for all of your chromosomes inside your cell, or for a plant, or for a fungus, or for a protist. And it's the thing that is one of the main differences between, say, bacterial cells and your cells, is that our cells have nuclei and bacterial cells do not have nuclei, right? So that's the difference between prokaryotic cells and eukaryotic cells, along with all of the other membrane-bound organelles. So the nucleus is a pretty darn big deal. It's sometimes called the chief executive of the cell, issuing orders. That analogy has never really worked for me. I don't like that one. I think of it as the central archive of the cell. It's the library of the cell. This is where we store all of the blueprints for making proteins inside of the cell. And the blueprints are stored in the form of genes. The genes are made of DNA, and they are stored in chromosomes. So one chromosome is a really long string of DNA, and one string of DNA can have thousands of genes strung together on it, just like a single manual might have instructions for many different parts inside of it. So one chromosome is kind of like a big book, which is a compilation of different blueprints for all kinds of different machines that you might want to make, different kinds of proteins you might want to make. But in their current form, the way we have it depicted here, the chromosomes are loosey-goosey. It's kind of like we took all the pages out of that book and we just, they're messed around. We made it all nice and loose. They're not in those tight little chromosome packages. You're used to seeing chromosomes as kind of this X shape. That's how you're usually shown them on like advertisements for DNA sequencing companies, or if you see it in like textbooks, they usually show you chromosomes in X form. But most of the time, your chromosomes, your DNA, is really loose and stringy. It's in what I like to call the spaghetti on the plate condition, where You know that you have individual noodles when you're having a plate of spaghetti, but when it's all together in the dish, you can't really count how many individual noodles you have. You just see one big mass of spaghetti on your plate. And when this happens in the nucleus, we call this substance chromatin. Chromatin is this mass of protein and DNA that you find inside of the nucleus here. The nucleus is a membrane-bound organelle, and in fact, it's got a double membrane. This double membrane is called the nuclear envelope. You can see that the nuclear envelope actually has pores inside of it, so there are little holes, little passageways to allow material in and out of the nucleus, and that's important because, again, this is a library, this is an archive. You need to be able to get information out of the nucleus. We want to keep it protected. We want to make sure it stays safe. We don't want any stray enzymes getting in here that would damage the DNA, because these are the original blueprints. We don't want anything to happen to them. We don't want any viruses to get in here that shouldn't, be coming in, because viruses, they want to get their own DNA into our archives, and we're very careful about who's allowed to keep their material here. But we do want to be able to use the information. We need to be able to make proteins sometimes, so we need to allow it out, and that's what the nuclear pores are for, to be able to get the information out occasionally. So we have the chromatin, which is the substance inside. We have the nuclear envelope, which is the container, and then we have the nuclear pores, which are the holes in the nuclear envelope. In the center, we have this dense little cluster here, and it's labeled the nucleolus. The nucleolus, the tiny nucleus, right? The nucleus inside the nucleus's friend, if you like. The nucleolus is a dense region of chromatin. It is an area where there is a higher proportion of chromatin than the rest. It's just a little bit tighter there. And what it is an overlapping region of various chromosomes. It's all of the parts of chromosomes that have genes that have to do with making ribosomes. The ribosomes are these little blue structures here. You can see them on the rough endoplasmic reticulum. This little dot, this little dot, this little dot, this little dot. Ribosomes are little machines that make proteins, right? They're the ones that take the instructions from the genetic material, and they actually read them and say, oh, okay, I see. This is how we would put together that protein. And we need to make some hemoglobin. We're going to make some heme proteins. Okay, this is how we would put that together according to these instructions. The ribosomes are the ones that do that. But the ribosomes themselves are made out of RNA, a nucleic acid, and protein. So we need to use the information inside the nucleus, the information inside the chromatin, to make ribosomes themselves. The nucleolus is where all of that particular information is stored. Now, I think I may have gotten away from myself there a little bit. The nucleolus is a dense region of chromatin inside the nucleus where parts for ribosomes are made. It's where all of the information for making ribosomes is going to be contained. It's an overlapping region from various different chromosomes, all kind of concentrated in one place. Now, one thing that's interesting about the nucleolus when we get to cell division, one step that occurs, one little footnote that occurs in one of the steps is that the nucleolus disappears, and that always sounded really exotic and mysterious to me. Where'd it go? The nucleolus just disappears. Where'd it go? And when I was a student, the teacher was always like, I don't know, it just disappears. But the reason it disappears is because it's just an overlapping region of chromosomes. So when all the chromosomes get bound up for cell division, well, there's no overlap anymore because they're all individual little packages. So it's just not there anymore. And then it reforms later when they become loose chromatin. So when we get to cell division, you'll see me mention that again. So that is the nucleolus and the nucleus and the nuclear envelope and the chromatin and the nuclear pores and the ribosomes even, but I'll talk about ribosomes a little bit more later on. Just outside of the nuclear envelope, and actually contiguous with it, meaning attached to it, really part of the same structure, you can see the membranes actually merge together, is the rough endoplasmic reticulum. And I mentioned in one of the previous videos, reticulum is Latin for net. So it's the net endoplasmic inside the cytoplasm, the endoplasmic reticulum, the network inside the cytoplasm. And you can see that it's this net-like structure. You have this series of chambers of cisternae going back and forth, snaking all around. And any information that were to come out of the nucleus, that information would be in the form of an RNA molecule. You might remember RNA from our discussion of nucleic acids. If that RNA molecule were to come out of the nucleus, it would be trapped inside this rough endoplasmic reticulum, and eventually it would be caught in this little maze here. It would bump into one of these ribosomes that studs the rough endoplasmic reticulum. It's called the rough endoplasmic reticulum because of those ribosomes, because it looks like it would be coarse in texture if you ran your finger over it. So, once it bumps into one of those ribosomes, that RNA gets read by the ribosomes, and a protein is produced based on the information in those instructions. So the rough endoplasmic reticulum provides a workspace for ribosomes, and they produce proteins, and then once those proteins are produced, it needs to be folded properly, and the rough ER can actually assist with that because inside this labyrinth are what we call chaperone proteins. You may remember that proteins, polypeptides, will fold themselves spontaneously into their three-dimensional shape, and that'll just be based on what the order of amino acids are in the polypeptide chain. You know, maybe these two amino acids happen to have a strong affinity for each other, so they'll be attracted to each other, and that'll create a little pinch in the chain right there. Well, sometimes there's a couple of different ways that the polypeptide might fold. There might be one right way and 3 or 4 wrong ways to do it. Chaperone proteins will prevent a polypeptide from folding in any of the incorrect ways, and therefore only correct proteins will be allowed to exit. The rough endoplasmic reticulum, which is this series of membranes, they take that protein and they put it into a vesicle. They pinch part of these membranes off and they put it into that little storage container, that little transport vesicle, and they send it off to the Golgi apparatus, which I mentioned in one of the previous videos when I gave you a tour of the cell. So that's what the rough endoplasmic reticulum does. It sits right outside of the nuclear envelope. It traps any RNA that makes its way out. It provides a workspace for ribosomes to do their thing, and once the ribosomes make a protein, they package that protein into a transport vesicle and they send them to the Golgi apparatus. Now, for some reason, the rough endoplasmic reticulum seems to get all of the glory, and its sister, the smooth endoplasmic reticulum, never seems to get due credit. So the smooth endoplasmic reticulum has a couple of important functions. Now, note, no ribosomes, that's why it's smooth. That is because the smooth endoplasmic reticulum has nothing to do with protein production. The smooth endoplasmic reticulum is all about the lipids. It makes lipids. It helps make phospholipids for more membranes. It helps make steroids and those sorts of things. It helps produce lipids and has another function as well. It helps detoxify circulating drugs inside of your system. So if you get a really good score on your biology test and you want to celebrate, and you celebrate all night, and you celebrate a little bit too hard, the next day you're not feeling so great, but the celebration is out of your bloodstream. The reason why it's out of your bloodstream is thanks in part to your smooth endoplasmic reticulum. You might think, it's like the liver of your cell, because that's what my liver does for me. But in fact, it's the liver of your liver as well, because liver cells are able to do their job partly because they have a higher proportion of this organelle inside of them. Liver cells have more smooth endoplasmic reticulum inside them than your typical body cell would. Again, form informs function. So you can tell a lot about the function of a cell based on how it's put together, what its order is, what its structure is. Lots of smooth endoplasmic reticulum, either that's a lipid-producing cell or that's something that helps detoxify the organism. Now on this next slide, you can see a micrograph of the rough and smooth endoplasmic reticulum. So here is the smooth endoplasmic reticulum, and here is the rough. All these little dark dots are ribosomes. So let's talk a little bit about ribosomes. Ribosomes are responsible for protein synthesis. Now, ribosomes are not membrane-bound organelles. And since they are not membrane-bound organelles, bacteria, prokaryotes, can have them as well. So you will find ribosomes in all cells. Ribosomes are one of those things you find in absolutely every living thing across the planet. Ribosomes are made out of RNA and protein, so no membranes involved in making a ribosome. Once again, their components are made in the nucleolus, but they are then assembled in the cytoplasm. You'll find them freely floating around in the cytoplasm, but also you'll find them fastened to the rough endoplasmic reticulum. And if we click to enlarge this picture, you can see that a ribosome comes in two subunits, and they kind of sandwich these other materials in between them. These two cyan structures that look like big boulders, these are the ribosomes. Don't be fooled, they're actually pretty teeny tiny on the scale of a cell, but we have the large ribosomal subunit and the small ribosomal subunit. The large ribosomal subunit is where the protein synthesis is actually going to occur. That is where the ribosome is managing all of the moving parts that are putting together the protein, putting together the amino acid, forming the peptide bonds and getting it all together. The small ribosomal subunit is bound to the RNA that came from the nucleus that's holding the instructions for making that protein. So ribosomes read information from the nucleus. They read the instructions from your genes, and they use that information to make proteins with the correct sequence of amino acids in the right order so that they will get the right three-dimensional folding so that they'll have the correct function. So all of those things we talked about in Unit 1 with the order of the amino acids and all of the bases of DNA and what order they go in and all of these things with like primary structure and tertiary structure of proteins, all of it is going to come up again because we're talking about how important the intricacies and interrelatedness of those ideas are. So make sure that you understand those ideas if you didn't feel too strong about them in Unit 1. Now, that said, I'm not going to spend too much time talking specifically about how ribosomes work, because when we get to Unit 4, we are going to spend a large chunk of time talking about the process of translation, which is all about how ribosomes put together proteins, how they read the information from genes and how they put together proteins. So for right now, what you're going to want to know is that a material called mRNA, that's messenger RNA, it's a nucleic acid. It comes from the nucleus with instructions for building a protein. And that is going to bind to this ribosome. It binds to the small ribosomal subunit, and then the large ribosomal subunit locks into place also, and that, based on the instructions on the mRNA, is going to join amino acids in the correct order, forming a polypeptide chain. And that means that we're going to make a polypeptide based on the information from the nucleus. Since all cells contain ribosomes, you can actually compare the genes for ribosomes between any two organisms in order to get an idea of how closely related they are. They're a really good benchmark for that. Now, that said, prokaryotes and eukaryotes do have a lot of differences in between their ribosomes. For one thing, eukaryotes have bigger ribosomes than prokaryotes do. Prokaryotes have what we call 70S ribosomes, whereas eukaryotes have 80S ribosomes. And we also have measurements of the size of the large and small subunits. Prokaryotes have a 30S small subunit and a 50S large subunit. And eukaryotes have a 40S small subunit and a 60S large subunit. Now, you might be looking at those numbers and saying, I don't think 30 and 50 add to 70, and I'm pretty darn sure that 40 and 60 don't add to 80. What's going on there? These measurements are in what we call Svedberg units, which are measurements of how quickly materials will come out of solution when we put them in a centrifuge. So it's not just a measurement of their size or their mass. It's kind of a combination of those factors, and some other properties could come into play as well. So that's why it's not a perfect relationship, because when you fit the two subunits together, they have a slightly different shape to them than the two individual units apart. So that's why it doesn't add up as neatly as you might expect them to. That part you definitely don't need to know for one of my exams in Bio 110. But you do need to know that the ribosome will deposit the completed polypeptide into the rough endoplasmic reticulum, which will then package that polypeptide into a transport vesicle. Where does that transport vesicle go? Well, it needs to go someplace where it can be modified and tagged and packaged and delivered to its final site, and that is going to be the Golgi apparatus. So that transport vesicle is going to leave one membrane-bound organelle, the ER, and it's going to travel to another membrane-bound organelle, the Golgi apparatus. The Golgi apparatus is a series of pocket-like membranes, kind of looks like a series of pita pockets sitting on top of each other called cisternae. And it has two sides. It has the receiving end, and it has the shipping end. What happens at the Golgi apparatus? Well, quite a number of things, actually. One of the main jobs that it has is that it takes all of the polypeptides, the chemical products of the cell that are coming from the endoplasmic reticulum, and it's going to distribute them and send them to their final locations inside of the cell. So maybe these proteins that the ribosomes made, maybe they're one of those channel proteins that allow materials through the cell membrane, right? Like maybe it's an aquaporin that we were talking about when we were talking about transport across membranes. So it needs to go into the cell membrane. The Golgi apparatus is going to send it to that final location. So part of it is like a post office, right? It aggregates all of the packages to one location and then it sends them out. It deploys them to their final sites. Another thing that happens, though, is that the packages get modified there. All the cellular products get modified there because sometimes you need more than one polypeptide to make a complete protein. Hemoglobin, for instance, needs 4 different polypeptides to be folded together in a particular way to make a functional protein. A lot of times, the multiple polypeptides will find each other here at the Golgi apparatus. Some proteins need to be modified as well. They need other kinds of groups attached to them. For example, a particular pigment molecule might need a specific kind of carbohydrate group slapped on in a particular location in order to do its job properly. That might get attached on here at the Golgi apparatus. So the Golgi apparatus receives, refines, stores, and distributes chemical products of the cell. And notice how tightly some of these organelles are connected together. The nucleus is bound by 2 membranes. That's a membrane-bound organelle, right? And that is surrounded by the rough endoplasmic reticulum and the smooth endoplasmic reticulum. That's a membrane-bound organelle right there. And they exchange materials between themselves. And then the endoplasmic reticulum, they pinch their chemical products off into vesicles, another membrane-bound organelle, and those vesicles get sent to the Golgi apparatus, another membrane-bound organelle, and then the Golgi apparatus, it receives, refines, restores, distributes. How does it distribute? Through more vesicles, right? So we have more membranes involved, and then those vesicles end up perhaps at the cell membrane, one very, very large membrane going around the entire cell. So this is what we call the intramembrane system, or the intramembrane network here. All of these different membrane-bound organelles. Now, these are not the only membrane-bound organelles. We still have to talk about the mitochondria and the chloroplasts, and like the ribosomes, mitochondria and chloroplasts are going to get their own section of the content where we take a deep, deep, deep dive into exactly how it is that they work. But when I say the difference between prokaryotic cells and eukaryotic cells is the membrane-bound organelles, that eukaryotic cells have membrane-bound organelles and prokaryotic cells do not, what do I mean by membrane-bound organelles? The nucleus, the endoplasmic reticulum, the rough and smooth, the Golgi apparatus, vesicles, vacuoles, mitochondria, chloroplasts. These are membrane-bound organelles.